everyone, how are you all? So myself Dr. Deeksha and I'll be teaching biology for CUET at only CUET. So guys, we will discuss about the previous year paper. Okay? You can see on my background like we have previous year paper displayed for 2023 biology, for which we will be discussing each and every question so that you get to know that what type of questions are asked in the examination and what is the approach to solve it. See, before I start with this discussion, I want to make one thing very clear. The only problem that we lack, right, the only thing we lack while studying or while attempting examination is we just try to mug up the things. Okay, we have to learn this, okay, this I do not remember. But you know, behind that, there is a little logic which is applied in each and every question with respect to each and every concept. So while I will be discussing this PYQs here, I want to bring to your notice that how and which type of concepts can you apply while solving the questions, even if you do not know that what is the answer to that particular thing. Right? So just let's come discuss and see that how this discussion can be fruitful to you guys. Let's start. Considering question number 1, what amount of energy would be at producer level if it is 50 kg at third trophic level? Now, if I see this question is of class 10th level, how there was a chapter called Our Environment in your Science NCERT textbook where you had a food chain. Okay, we studied the concept of food chain which started with producers, which constituted the first trophic level, going to the second part, that is second trophic level might be the primary consumers, then the third trophic level secondary consumers going above, right? And the food chain mostly comprises of 4 to 5 animals or organisms only, right? So now they are telling that at third trophic level we have 50 kilojoules of energy. Just use your mind and go in a flashback. There was a law called as 10% law. This 10% law demanded that the producers transferred 10% of energy to second trophic level, then they transferred 10% of energy of the remaining to the third one, and then so on, right? So if this trophic level has 50 kilojoules of energy, that means second trophic level had more than third, that is why its 10% went above, correct? So 10, 50, adding one more 0 comes out to be 50 kilojoules, right? Similarly, first trophic level would had more energy, right? So adding one more 0 to it, that comes out to be 5000 kilojoules. So here the correct answer lies to be 5000 kilojoules. Now what did you understand from this? That the question had a very simple concept and it was based on your previous knowledge only. But it is just your presence of mind should be there in the examination. You should have a deep interest in the subject to solve the same. Correct? Now moving on to the next question. This question always possesses a problem for the students because this is a learning thing. But even if you do not know anything about it, one thing you always know is that eyes potato. We have been always reading about that potatoes have some white spots or buds over it. We call them as eyes from which new potato can arise. This you have studied from your junior classes. And even if you have not, you have an idea about that the potato bears some white outgrowths which you can see in your daily life as well. So that only happens, these are basically the structures which are the vegetative buds of the plants. Because plants show two types of reproduction, asexual and sexual. And in asexual, they show vegetative propagation. Correct? So for vegetative reproduction, they need to have some outgrowths from which the new individual can develop. So what type of an outgrowth they have? Let's study. Potato has eyes. We already know about it. This is from our junior knowledge. Then rhizome is some structure which is growing underground and we always refer ginger as an example for that. Offset is seen in water hyacinth, where this plant has a parallel stem originating, and then from that a new plant comes out. So this is all vegetative propagation. Okay. Then bulbil is seen in agave. So matching all the options, A is part 2, so we have this. Okay, A, everything we have part 2, B is 1, so we can figure out from B and C. Then C is 4th, this, and D is 3rd. So D part is probably the correct answer. Okay, so this was based on asexual reproduction in plants, that is vegetative propagation. Then coming next is a very interesting question. Anybody who is lacking interest in biotechnology, just please wait. I'll be posting lectures on biotechnology where it will be very simple and easy for you to how grasp biotechnology, right? So here just reading one question out of it is copying genetic information from one strand of DNA into mRNA. Now what happens is DNA is our genetic material and we all know that it passes over generations to generations. But what is actually DNA carrying? DNA is carrying some sort of information which is present with respect to our body features, right? Now that DNA has information in the form of proteins. So how does it get converted? So basically the DNA it gets modified into, the DNA is translated into mRNA. Then mRNA is read as proteins and proteins contain the information of that specific feature. So here copying genetic information from one strand of DNA into mRNA, what can we name it as? Quickly, I just told you what can we call it as? Transcription. Okay, so we have the first answer. Then going to question number 4, transfer of pollen grains from anther of one flower to the stigma of flower of different plants. Now you must have read in your previous classes about the pollination concept, where it was read in junior classes regarding self and cross pollination. So self pollination was when the anther from the stigma of the same flower was going to the pistil of the same flower. But when it is of the different flower or different plants, we call it as the cross pollination. So here it is of a different plant, we will name it as xenogamy. This is the name given to that particular thing. Coming out to question number 5, if we say homozygosity. So homozygosity is what? Homozygosity is basically pure lines. You must have studied Mendel's experiment in class 10th, where we had genes such as T, T and small t. This was the first cross which Mendel did in the pea plant, hybridizing tall pea plants with the dwarf pea plants. So when he took the genotype of the same, he took it as tall TT into short TT, like capitals and small letters. Okay, so homozygosity is this, that you have both the alleles same, right? So whenever these alleles are different, for example this is heterozygosity. So these where we have similar alleles, we call them as pure breeds. That means they have no mixing of alleles, right? So homozygosity means pure lines. Then talking about the next, so A 4th, we will omit C part because A is first, right? One option is already deleted out of 3. Then outbreeding. What do you mean by outbreeding? Outbreeding is basically cross between the different breeds, okay, and it overcomes the inbreeding depression. So these two words and these two words are very interrelated to each other, where outbreeding is something when we are crossing two different breeds. But when we are overcoming their inbreeding depression, that means we are continuously hybridizing the same family species. So that will lead to accumulation of genes and might cause inbreeding depression, that each allele would not be able to express itself. So we need to have some variation for better survival. In that term, we prefer doing outcross by hybridizing it with some native species, so that a new species, new gene is introduced in that gene pool, and and the inbreeding depression is overcome. Right. So basically we are avoiding the family transfusion and we are just bringing some native species so that we have variation in our genetic pool. Okay. So now we have got two answers, outbreeding, B third. So we have this. Okay. Hissardale. This is a species which was developed by crossbreeding. Okay, so anyways it was the leftover one. If you know the concepts of the other three, you can easily determine the answer. So B part lies the correct answer. So we have fruitfully discussed question number 1 to 5. If you are having little knowledge also of the subject, you are good to go with these questions. Now moving next. Okay, this is something which is called as that you will have to learn. You will have to, a factual question this is. What is the question? How many episodes of mass extinction of species have occurred on Earth since origin and diversification of life? So this is a very factual question. 5 mass extinctions have been seen on the Earth since the life has originated. Okay. Moving on to the next question number 7. Arrange the steps of polymerase chain reaction in proper sequence. What is actually PCR? Why are we performing this? We are performing this to study some concepts genetically and very closely. Now for that, what do we have to do? PCR is basically used for diagnosing, but right, in biotech, in our studies, we are basically using it to study the concepts. Now PCR in proper sequence can be arranged as, first is selection of desired fragment of DNA. You will obviously choose first the desired fragment, then you will have to denature it, right? You will have to get the original thing out of it. Then you will anneal it, then you will extend it, and finally you will amplify it. So these are the basic steps that a PCR offers, and the details of which will be given in your chapter of biotechnology. But it is right now telling one thing only, that we are studying one fragment of DNA. While we are taking it, we are first denaturing it, we are then annealing it, we are extending it, and we are zooming it and see. We are amplifying it. Correct. So based on these steps, I think A part would be the correct answer. Just match once. Yes, correct. So A part is the right answer. Next, process of evolution of different species in a given geographical areas as starting from a point and dispersing to others. That means they are saying that they have a common ancestor starting from the point and then having multiple more species. So that case is seen in adaptive radiations. Now these are the concepts of chapter evolution, which are telling us that when we have a common ancestor and from those a few species are arising, that is the concept of adapting radiation. Moving to next, identify the mammals. Here is the word you should know. We have to select the mammals which live wholly in water. So dolphins and seals we are very sure because we know them in our real life also. Whereas seahorse and sea cow, there is only seahorse. Why? Because it's not a mammal. It's an osteocyte. You must have read in class 11th Animal Kingdom. Okay, even if not, you will memorize it. Fine. So dolphins, seals and cow are the mammals which live in the completely in water. So omitting option C, I have only D part. That's my correct answer. Correct. Next. Okay, very interesting question. Human protein alpha-1 antitrypsin. Now this is a human therapeutic mechanism. This enzyme is a human therapeutic means where our body is itself trying to therapy its own body, where our body is itself becoming the medicine to certain disease. So this is actually used to treat a disorder called as emphysema, and this is also a very factual question based on the studies, right? Next. In a laboratory, population of 80 fruit flies, 8 died in a week. Then the death rate of fruit flies is what? Okay, so death rate is basically, death rate is number of individuals died upon total. Now here died are 8, total are 80, that comes out to be 1 by 10, so 0.1. 0.1 individuals per fruit fly per week. So C part will lie the correct answer. And hence we are done with question number 1 to question number 10. Moving to the further page and analyzing what more questions have been given in the examination, let's move on to question number 12. The A and B polypeptides chains in insulin are linked together by which bond? Here the key word is insulin. Insulin is a hormone produced by pancreas in human body, which we have been studying, and we are very well aware about the disease it causes. But now insulin, being a hormone, is a chemical and must be having some chemical structure. So in your book NCERT, it is mentioned that A and B polypeptide chains of insulins are linked together by disulfide bonds. Okay. Then question number 13 is saying that the karyotype in Klinefelter syndrome, it's a syndrome called as a Klinefelter syndrome, where while the baby is being formed, while the genotype of the human baby which is formed is being determined, there one X chromosome is extra. Okay, normally what do we have? Normally we have 44 plus XX in case of girl and XY in case of boy. But in Klinefelter syndrome, we have 44 plus XXY. We have one X chromosome extra. So here the answer will be C part. Everybody is aware that human cell has 23 pairs of chromosome. That means 46 chromosomes. Then question number 14. In some species of Asteraceae and grasses, seeds are produced without fertilization. Now see, this is a process where seeds are produced without fertilization and we call it as apomixis. You must have read in your reproduction and flowering plants that mostly flowers are produced are seed ones, but many are produced without fertilization. Right, they have seeds, but those seeds are not produced from fertilization by the fusion of male and female gamete. No, they are produced without that fertilization, and that concept is referred to as apomixis. Next, fine powder of recycled modified plastics. That means we are actually blending the plastics, we are actually mixing it. Okay, so the mixture which is obtained by recycling is the poly blend. Then arrange the following steps for replication of HIV in the correct sequence. Now we all know that what is HIV. HIV is nothing but referred to as human immunodeficiency syndrome, shortly named as HIV, responsible for causing AIDS. AIDS is acquired immunodeficiency syndrome. Even if you do not know, kindly memorize it. That's why I have written it completely. Now, what are they asking here in this question is that how does this HIV virus in our body replicate? Basically, we have studied from our past knowledge that this virus is very clever. It does not have any of its own machinery. So, basically, it gets into the cell of our. Now, their first thing it does is infection of a normal cell by virus. It is invaded into our normal cell. then it produces its viral DNA. It hampers our DNA and takes over the control and starts producing its own DNAs. Now from that DNA, new viral mRNA is generated. New viral RNA is generated by the same infected cell which the virus has captured. After it has generated, it will start producing new viruses. This is how it will replicate and produce more and more of its own copies. So the correct sequence is B, then C, then A, then D. So D part is the correct answer. Next question. Question number 17 is saying, identify the free living nitrogen fixing bacteria. The free living nitrogen fixing bacteria is one which is not dependent on any other organism and it is living freely on its own. Like for example, rhizobium is located in the root nodules of the high leguminous plants. Right? So it's not a free living, it's dependent on the leguminous plants, roots nodules of there. Right? Whereas Azotobacter and Azospirillum, these are the bacteria which come under the category of free living species. So we'll call them as C and D as the correct answer. Now if you see, we have discussed 17 questions till now. With the all question concepts are coming directly from your NCERT. So the golden and the key rule is to focus on the same. Correct? Then question number 18 is saying that the backbone of polynucleotide chain is formed by what? Now I tell you that this polynucleotide chain, this means formed by multiple nucleotides. Nucleotides are one which are made up of sugar, phosphates and nitrogenous bases. Three are the components which form this nucleotide chain. Right? Now this nucleotide chain has a backbone where it is adjoining the other. But in that backbone, it lacks nitrogenous bases. So it only has sugar and phosphates. This is the concept which you study while you are understanding about the polynucleotide chain. Then chromosomes in neocytes of butterfly. This is a factual question. Butterfly has 380 chromosomes. This you have to learn. We'll see the option. Just a second. Yeah, we have it, option number D. Okay. Then question number 20, the enzyme which is used to remove nucleotides from ends of DNA fragment. That means we have a long strand of DNA and we want to remove some fragments. So for that an enzyme is used which we call as restriction exonuclease. Exo means remove out. Nuclease means nucleotides, okay, and restriction is used for the name word enzyme, right? So restriction exonuclease. Then spermiogenesis is the process. Now basically spermiogenesis is the formation of sperms, but sperms are formed from spermatids. Before spermatids we have primary spermatocytes and then we have secondary spermatocytes. Spermiogenesis is the process where the sperm is being formed finally and the sperm is formed from spermatids. So A part is the correct answer. Direct NCERT questions. Very, very important. You have to grasp your book very nicely. Then match list 1 with list 2 as per question number 22. Typhoid, very, very easy question. You know, it is the best way to memorize this. Match it. Pneumonia, phyllis. Common cold is caused by rhinovirus. This we have been learning since our junior classes, and even if you do not know it now that common cold is caused by rhinoviruses. Whereas elephantiasis is a disease which is caused by Wuchereria malayi. Now elephantiasis is a disease where our foot increases in size massively. It forms such a big foot. Okay, so the options are becoming A2. So we have, it can be A, it can be D. Pneumonia B3, so it is going with D. C1 and D4. D part is the correct answer. Next, colostrum. What is colostrum? Mother's feed. okay, for the baby, contains that milk is known as colostrum, which is filled with antibodies, IgA, very good for child's immunity. So that is why it is said that a newborn baby should be fed mother's milk, because it contains some important things which will help it living a healthy life. Okay. Next, identify the common sexually transmitted diseases. Now, if you see these options, you will be easily able to identify syphilis. For say you do not know what are the other diseases, you omit the things where influenza is present, because influenza cannot be a sexually transmitted diseases. Then elephantiasis is not a sexual transmitted diseases. So A and B, both the options are out. Now, C part and D part is left. C, again we have elephantiasis out. So we are only left with D part, and now if we look at it carefully, we will not get a disease which we know about except from syphilis. So, and the student who has learned might know that chlamydiasis and trichomoniasis are also STDs. So D part is the correct answer. Next, what kind of IUD is Lippes loop? Before understanding Lippes loop, you need to understand what is IUD. So IUD is nothing but intrauterine device. Intrauterine device is a contraceptive method to avoid pregnancy. Right. So this is a device which is used by the people. They go to the doctor, get introduced in the body of the female, and due to which, so that the sperm does not encounter with the egg. Okay, but these IUDs are of various type, and out of which one type is Lippes loop. Now Lippes loop has the major drawback that it is non-medicated. Okay, and these IUDs are otherwise not recommended for contraception, as it is very unhealthy for the female's body. So we have non-medicated as the best option here, because medicated cannot be possible. Since it is non-medicated, it won't be having any chemical as well. So A only is the correct answer. Right? I told you, use your mind conceptually. Okay? So we are halfway done. We have covered question number 1 to question number 25. I hope you are gaining knowledge out of these questions, and you are able to figure out that what type of questions are asked in the CUET examination. Now, question number 26. Signals of parturition originate from which of the following? What is parturition? My question is, what is parturition? The parturition is the process of delivery of the baby. Like when the body is giving signal that the baby should be delivered out, that is parturition. But those signals are originated from two things. One, the fully developed fetus. Once the fetus is fully developed, it will give an ejection reflex that I want to come out. Okay, and the other one is the placenta. So we have C and D as the correct answers. These two things contribute majorly that the parturition takes place. Okay. Question number 27. Phenylketonuria is associated with what out of all? Okay, so I'll tell you, phenylketonuria is basically pleiotropy. Now, what is pleiotropy? When one gene is used, okay, for multiple things. Whereas hemophilia is something which is sex linked. That means it can be transmitted over generations since it is linked to sex chromosomes. On the contrary, snapdragon is an organism that shows incomplete dominance. Incomplete dominance means we have partial expression of both the alleles. Okay, so what happens is if two alleles are mixed, a hybrid comes out. Okay. Dihybrid cross we all know from class 10th only that we have 9:3:3:1 ratio. So A3, C and D, B4 only D, C1 and D second. Done. Correct. Moving to next question, question number 28. Which of the following is not a technique of genetic engineering? Which is not a technique of genetic engineering? Now I'll tell you, genetic engineering is something where we are using it for, like example, I tell you, where new products are being developed, like for example creation of recombinant DNA, okay, use of gene cloning, you are cloning, then gene transfer, you are transferring it from one to another, right? But grafting is a method of artificial vegetative propagation. So it has nothing to do with genetic engineering. So here D part is not a technique for the same. Question number 29. It is asking something about yellow mosaic virus. Yellow mosaic virus. What is yellow mosaic virus? If you go in a flashback, you will be amazed to know that in your class 8th, you had a chapter called Microorganism, Friends and Foe. There in Microorganisms at Foe, you had studied a plant disease called as yellow mosaic virus. It is caused by yellow mosaic virus and we called it as yellow mosaic disease of bhindi. So okra is the scientific name of bhindi. Even if you are not aware of anything else in the question, this yellow mosaic virus will give you a clear hint that it is related to okra. See, I am teaching you various methods also. So it is not always compulsory that you remember each and everything in the examination. But sometimes it's very important to use your past knowledge and use your mental tricks. You should have your presence of mind to clickbait to the question. And that's what has happened here. We had just linked yellow mosaic virus with bhindi. And that's the okra. Okay. Moving on to the next question, that's question number 30. This says that in which ecosystem a large fraction of energy flows more through DFC than through GFC. Before we have any remarks about the question, we need to know what is DFC and what is GFC. Now I'll tell you that DFC is detritus food chain, the one which starts with decomposers. That means normal food chain, which is the GFC, okay, it has producers in its first level. Okay, we have producers here, but detritus food chain DFC has decomposers, and that's mostly seen in the land ecosystem. Now, if you see the options, there is only one land ecosystem here, that is the forest ecosystem, where the cycle is starting from the decomposers. Okay. Question number 31. Discuss diseases caused by fungi are. Sorry, I mentioned discuss wrong. The diseases caused by fungus. You know, fungus has very specific diseases. Like when I am seeing the options, I will remember brown rust of wheat. Because brown rust of wheat was a very that disease which I remember from my past knowledge. In class 8, this disease also has been mentioned in the book. Brown rust of wheat caused by fungus. Apart from this, we have red dot of sugarcane and late blight of potato. Tobacco mosaic is caused by virus, and I think black rot of crucifers is by the bacteria. So we have correct option as B, D and E, B part. Correct. This was regarding this question. Moving ahead, question number 32. Okay. See, I will tell you the reality what happens. Whenever we see such a question in the examination, students tend to omit it. Students tend to leave the question and move it to the next, because such difficult words, do not align with the concept. But I'll teach you a good method by which you can, at least attempt the question, right? See, If you are not able to see anything over here, you can see ELISA and you can understand molecular diagnosis. ELISA is what? ELISA is a diagnostic procedure nowadays. So here is the word diagnosis, molecular diagnosis. You can immediately link it. So we have A, third. Now if you see from the option, we only have two choices, A and B. So C and D are already cut out. Elimination method. Now, next thing, I told you in this paper only previously when we were talking about human alpha-1 antitrypsin, this one, that it is a human therapeutic procedure where the body itself is producing an enzyme to treat emphysema. Okay? So, this is directly being linked with each other, human therapeutics as alpha-1 antitrypsin, correct. So, we can have B2, that is A part, then validity of GM research. Now, whatever researches are have been made genetically is being approved or is being validified by an organization called as GEAC. So, D first, and transgenic Rosie was a cow that was produced from human alpha lactalbumin to produce better milk. Okay, so C fourth. So A part is the correct answer. Now basically what I wanted to tell here was that we had already eliminated C and D options. Correct, and we had to filter out only from A and B, which I just used my common sense and did it out. Okay, so see, firstly you should know the concepts, that is the priority, but even there is a lot of stress during the exam, there is everything, and you had such a bunch of syllabus to study, so you tend to forget things. At that time we should use these tricks and methods to come to the conclusions. Okay, see next. This is one question also which everybody tends to leave because this is based on the names of scientists. So, okay, I will not give any conceptual knowledge over here because it does not have any, it is just the facts. Basically, one thing which everybody studies is that this scientist Erwin Chargaff had always told us about that the nucleotide sequences number are equal. So, we can always link directly C fourth part. And if you see here, we have again A and B options, C and D are already eliminated. Apart from that, we know that Jacob and Monod have made the operon model, and Alex Jeffries is used as for DNA fingerprinting. You might be thinking, no ma'am, we do not know this much also. So, if you do not know that much, you must have been aware about this because this is written everywhere in your book. Okay, so Alex Jeffrey, DNA fingerprinting, Jacob Monod, operon model. So, I think B would be the correct answer. Wilkins and Franklin, X-ray diffraction data DNA, B part is the correct answer. This is the direct question. See, here I cannot give you any conceptual explanation. This is the fact thing which you should know. Okay. Then question number 34, it says bone structure of 4 limbs of vertebrates like whale, bat, cheetah and humans are evidence of what? Now if they are specifically talking about bone structure, we click one thing, that either these bone structures might be having similar structure or they might be having similar function. Now if I talk about the species whale, bat, cheetah, human, one is aquatic, one flies, the other one stays, the other one is a fast runner. So obviously they must be having different functions. So this implies to me that they must be having same type of structure but utilized for performing different functions. So such case is studied under divergent evolution, where you have same structure but the functionings are different and from a common thing many have been originated. Correct? Question number 35, what of the following team refers to the confinement of species to specific areas and also the same species not found anywhere? That means they are referring us to species to a specific area. Go to class 8th, remember conservation of plants and animals chapter, and there you had read about endemic species. Endemic species are the ones which were found in one particular area only. So endemism, it refers to the confinement of species to that particular area. Then jhum cultivation. Jhum cultivation is the other name of slash and burn agriculture, where we are burning the land. First we are clearing it, that means deforestation is occurring, then we are burning it, we are allowing the soil to rejuvenate, and then we are again planting. So this has now been banned actually, but earlier it was being practiced in northern eastern states, which was actually causing deforestation. A lot of loss to the flora of the eastern region. Okay. Finishing one more page and moving on to the next. Slowly and slowly we are proceeding towards the end of PYQ discussion. Question number 37. Seeing the chart and table, I can figure out it is related to biogas production. Now, if I see the chart, for example, you are not able to memorize that what actually was the biogas production procedure. But if you are looking at the chart and you will see the words primary, secondary, activated sludge, biogas. So, we get to know this much that biogas is the final product. And this final product was obtained in anaerobic sludge digester. Because last anaerobic things are left, that is how biogas was being produced. Correct. So, A fourth we are clear. And we have only option A and C. Now, secondary treatment always involved change in biochemical oxygen demand, because in primary treatment what has happened, we had segregated the primary sludge, and due to mixing of air, a lot of aerobic bacteria had sucked up all the oxygen. So, the oxygen demand went less. So, that was change in biochemical oxygen demand. Finally, the bacterial flocks were introduced in the activated sludge, and at last the anaerobic sludge produces biogas. So, seeing the options, we know the answer is B part is 3, so C part. C 1 and D second. Absolutely suitable and correct options. So, I think and I hope that you people are able to understand whatever I am speaking here, and I am telling you the best mechanisms you can follow to filter out the questions. Then, which of the statements are correct about thalassemia? Thalassemia alpha, thalassemia beta is actually caused due to the changes due to the problems in alpha globulin, beta globulin. Okay, but the first says that in beta thalassemia, production of alpha globulin chain is affected. So this lies wrong, because beta thalassemia is named beta, that is why only, since the beta globulin chain is affected. Okay. Next, alpha thalassemia is controlled by two closely linked genes, HPA1 and HPA2. Yes, correct. So whenever we will study about thalassemia, I will tell you in detail. The genes HP1 and H2 are present on chromosome 11 of each parent. No. Beta thalassemia is controlled by single HBB gene. Correct. It is located on chromosome number 6 of each parent. No. So basically B and D are the correct answer. This is a total conceptual and factual question. You will have to learn that what are the types of genes and on which chromosome they are present. So here we can do nothing as using our mind. Here this is, if you know, you know, if you do not, you cannot. That's the thing. Okay. Question number 39, the organism responsible for large holes in one Swiss cheese. Okay, so do you remember that while we watch Tom and Jerry, we have that cheese and it has a lot of holes in it. Okay, so that is nothing due to the organism, that is Propionibacterium shermani. This causes fermentation and thus we see those holes because a large amount of CO2 gas evolves from there. Okay. Then interaction between two species where one species benefited and the other one is neither benefited nor harmed, that is commensalism. This is also something you should have learned. So you must be aware about it. Like predating is hunting, parasites is affecting the other. So like that you should have knowledge about it. So here the answer to this question is commensalism, where one species benefited and other is neither benefited nor harmed. Correct. Next, direction for question number 41 to 45. We have finished this type of questions. I think we are only left with 10 case study based questions now. So just let's quickly read the passage carefully and then we'll attempt the questions as per it. I hope it's visible to you guys. Sorry. Read the passage carefully and attempt the questions. In our body, cell growth and differentiation is highly controlled and regulated. They are saying in our body, the cell growth and differentiation. Differentiation means the cells are being differentiated into or more structures for specialized functions. Is highly controlled and regulated. It is in a deep control. But in cancer cells, there is breakdown of these regulatory mechanisms. Anyhow, during cancer, this problem is faced. Cancerous cells just continue to divide, giving rise to masses of cells called tumors. Cancer cells lose this regulatory control and they keep on dividing, and those extra cells are named as tumor cells, which are invading other tissues and causing problems. Benign tumor normally remain confined to their just a second. Yeah. Benign tumors usually remain confined to their locations. Yes, we have two types of tumors, that is benign, that is malignant. Benign is one which are localized, and the malignant are one which keeps on spreading. So, malignant tumors on the other hand are the mass of proliferating cells. These cells actively divide and starve the normal cells by competing for vital nutrients. These cells will actively divide very quickly and starve the normal cells, and they will remain the normal cell lack of nutrients. Cells slowed from such tumors reach distant site through blood. Now the extra cells will travel through blood, and wherever they get lodged in a body, wherever they are fixed, they start new tumor over there also. They start replicating, more and more cells produce more and more tumor cells engaged. This property is called as metastasis, but this is the property of malignant tumor. In benign, it remains confined to that area. So, transformation of normal cell into cancerous cells may be induced by any physical, chemical or biological agent, and these are termed as carcinogens. Okay, carcinogens basically do it. Now, the statement which is correct for cancer cells are that these cells grow very rapidly, invade and damage the normal tissue. These cells grow very slowly, wrong. These cells grow very slow, wrong. Very rapidly, and when liver cell only, no. Whatever they feel, wherever they get lodged, there is no specific site. They just go there, they live and they damage. Okay. Cancer is caused by carcinogens and oncogenes. So these are the two words which are used for cancer. So the answer is here A and D. You must have seen the cancer department written as oncology department, right? And cancer being termed as carcinoma. Okay, so these are the two words which are used for cancer. Question number 43. Normal cells show a property that does not allow their uncontrolled growth. Now what happens is, some normal cells show a property in which they do not allow the other cell to enter or come in contact with them. So that property is known as contact inhibition. That will not allow its uncontrolled growth. It will remain confined. So normal cells show this property of contact inhibition. Then benign tumors are normally confined to original location and do not spread to other parts of the body. They do not spread. Whereas in all other options you will see that they spread, but they do not spread. Okay? Tobacco smoke has been identified as the major cause of lung cancer. Daily we are seeing so many ads. You go in a PVR, you have a tobacco ad running. They are using the word फेफड़े, that means lungs, and they are highlighting it again and again that tobacco is damaging your lungs. Okay, so tobacco is responsible for the lung cancer. It's a very bad carcinogen. Okay. Okay, the study of cancer was quite depressing. And it is a condition actually which is affecting a lot of human population nowadays. The lifestyle issues and many other complications are just leading to cancer in early ages of life to the people. So we should, live a healthy lifestyle, and moreover it's just the error of genes. Actually I say that when the cells, normal cells, start to producing tumor cells, Okay, but yes, cancer is one thing which has been leaving a very bad impact on the population, and it ultimately leads to worsening of the body and lasts in death only. So that's the sad reality over here. Now if I talk about the another and the last case study passage given in your this paper, you can read it very nicely what it is written. An overwhelming majority, 99% of animals and nearby all the plants cannot maintain a constant internal environment. Like we people have a constant temperature of 37 degrees Celsius. We are warm-blooded animals, okay? We are maintaining a constant temperature, whereas majority plants and animals, they have written 99% cannot maintain, okay? So their body temperature changes with the ambient temperature, means with the outer environment, their body temperature keeps on changing. Their body temperature changes with the ambient temperature. In aquatic animals, the osmotic concentration of body fluids change with that of the ambient air and water osmotic concentration. Aquatic animals, they keep on changing with respect to their aquatic environment. These animals and plants are simply called as conformers. What do we call them as? Conformers. Considering the benefits of constant internal environment to the organism, we must ask why these conformers had not evolved to become regulators. Regulators are the one which regulate, okay. So, considering the benefits, we must ask why these conformers did not regulate into regulators. Sorry. Recall the human analogy we used above. Much as they like, how many people can really afford air conditioner? Very low. India is full of people who are poverty stricken. So very low strength. Okay. Many simply sweat it out or resign themselves to suboptimal performance. Mostly population is sweating out. Okay. In hot summer months. Okay. Then very small animals are rarely found in polar regions. In polar regions we have very पोलर रीजन्स में क्या होता है ना यार कि बहुत ज़्यादा स्मॉल एनिमल्स कम मिलते हैं, ठीक है. वील रीड द रीजन. ड्यूरिंग द कोर्स ऑफ एवोल्यूशन द कॉस्ट एंड बेनिफिट्स ऑफ मेंटेनिंग इन अ कांस्टेंट इंटरनल एनवायरनमेंट वर टेकन इनटू कंसिडरेशन, वेयर सम स्पीशीज हैव इवॉल्व्ड द एबिलिटी टू रेगुलेट बट ओनली अ लिमिटेड रेंज ऑफ एनवायरमेंटल कंडीशंस. लाइक दे कैन रेगुलेट बट ऑन टू टिल द एक्सटेंट ऑफ सम एनवायरमेंटल कंडीशंस. इफ इट इज एक्सटेंडेड बियॉन्ड दैट दे कैन नॉट. ओके. They turn into conformers again. If stressful external conditions are localized or remain only for a short duration, the organism has 5 other alternatives for survival. So this is a concept which is actually based on conformers and regulators, where they are telling us about the difference, that how they are not sustaining as per the outer environment. Question number 46 based on the same says, conformers have not evolved to become regulators. Why? Because conformers are generally small sized. No, their cost of becoming a regulator is very high, because they have to use a lot of energy. So the cost of regulation is very high. Identify the animal which is not a conformer. So if you see here, hummingbird, shrew, polar bear, lizard bird. Polar bear is not a conformer, it is a regulator. It lives in a Antarctic region. It has to be a regulator, okay. Conformers are the animals whose body temperature changes with the surrounding environment. Like we regulators do not change with the surrounding environment, okay, but they keep on changing with respect to the surrounding one. Identify the set of aquatic animals whose osmotic concentration is not constant. There might be some aquatic animals where the osmotic concentration is not common. Now if you see the examples, whale, fish, mammal, vertebrates, whale, dolphin, fish, there the osmotic concentration is constant. Why? Because they are large, huge organisms, okay, they are maintaining it. But amoeba and paramecium, being the unicellular organisms, fail to do so. So there the osmotic concentration is not constant. I hope you are aware about the word osmosis, where the balance should be maintained between the solute and solvent in both the conditions. Oh, we are proceeding on to the last question now, question number 50, and we'll be done with the PYQ discussion. So, identify the statement which supports the reason why small animals are rarely found in polar region. Actually, what happens, I was going to tell you there also, that small organisms have a larger surface area in respect to their volume. Okay, because since they are small, the volume is less, but the area exposed to the environment is large. So, in that case, they tend to lose more heat. And that is why they are not found in polar regions, because we require such organisms in polar region which have the ability to conserve energy, which they have the ability to conserve the heat, so that they remain warm and can survive in the pathetic cold environmental conditions also. Okay. So, first option is correct. Then we have small animals have larger volume. No, this is wrong. Shrews and hummingbirds are not found in polar region. Correct, because they cannot be found in the polar region. Then small animals have smaller surface area. No, this is wrong. So, A and C, with respect, we have C part as the correct answer. Okay. Now we have gained a lot many things. We have studied about a huge number of questions that appeared in this examination, right? You can see we studied about this case study. We had this case study based on the cancer patients, cancer disease. Okay, we studied about thalassemia, an important concept. We studied about commensalism, amenzialism, predation, etc. We have biogas production issues. Then we can have read about the jhum cultivation. We had major questions from biotechnology also, where we are reading about different scientists, what are they causing, right, their discoveries. We had some questions related to ELISA, some related to PCR. Okay, important points I am telling you continuously. Then diseases caused by fungus, virus, bacteria, this all should be clear. Apart from that, we have some information regarding the food chains, detritus food chains. Okay, then we have yellow mosaic virus, again disease caused. Diseases are very, very important. We are having many questions where diseases are being asked on, right? Then Mendel cross, evolution and heredity, we are having questions. Human parturition. Next, if we see, I am just making you revise that what topics the questions are asked on. You can see IUDs, then sexually transmitted diseases, colostrum. Colostrum, you know, when we make you people study about colostrum, then also it is said that it is very, very important. So, you see any of an examination, there you will find colostrum. Okay. Match the columns, maybe also we had the organisms causing particular diseases, and spermiogenesis. We had question from human reproduction also. Same enzymatic biotech questions, chromosomes, backbones of polynucleotide chains. So we had majority of questions from there. Then free living nitrogen fixing bacteria. I mentioned that bacteria, virus, fungi you have to learn, very, very important. HIV virus, recycled modified plastic, we had one question only from the same. Then general term like apomixis, Klinefelter syndrome, insulin chain. Here if we see, we had question from food chain, we had question from death rate, human alpha-1 antitrypsin. This is also said to very, very important. When we teach in the class, we refer that this is very, very important. Some mammals, some evolution stories, right? PCR we've already studied. Okay, Mendel's laws and experiments. Some related to cross between the species, copying genetic information, pollination, and some vegetative reproductive structures. So summarizing everything, we had good amount of questions from class 12th biology, and we have been covering the best part. All of this has been covered in the NCERT. You just have to read good enough in the whole book in an year so that you get to pass through each examination. Right. So that's all with the PYQ discussion, and I hope you have understood and gained some knowledge on how to attempt the paper, and must have also got an idea that what type of questions are asked in the examination. So that's all from my end. Thank you so much, and thank you. That's it.